Now, boys and girls, welcome to the second segment of this video. Before I just got cut short here. I want us to quickly talk about how, what happens after that. What happens is that tRNA carries specific amino acids to the ribosome, right? When the anticodon of tRNA matches the codon of mRNA. So thus, this means that in this case, tRNA would be giving us would be holding an adenine. It would also be, and then from there on, because of cytosine, it would be having a guanine. Because of guanine, it would be having a cytosine. And then, yeah, because of adenine, it would be having a uracil, because we're dealing with RNA. And RNA only has adenine, guanine, cytosine, and uracil. It does not have thymine. Now, that being said, tRNA brings required amino acids to the ribosome, and the amino acids are bonded to each other by what we call peptide bonds. Peptide bonds. Now, when 50 or more amino acids are joined together, a particular protein is thus then formed. Now, this process can go wrong, and when this process goes wrong, this is called a mutation, boys and girls. It could be good, it could be bad. Okay. Now, what is a mutation? Now, the definition of a mutation is a change in the nitrogenous base sequences. Okay? And we have two types of mutations. We have a gene mutation and a chromosomal mutation. So a gene mutation usually occurs during protein synthesis. A chromosomal mutation would happen usually when we're dealing with two different, two different two different gametes, right? And in this case, for example, chromosomal mutation could be what we call aneuploidy or polyploidy, and we could also have what we call let me just quickly think about it, yeah? Down syndrome, yes, trisomy 21, where there's an extra chromosome or a missing chromosome in a certain aspect, but we'll get to it just now. And I just want to give us the format for answering mutation questions, boys and girls. Now, when answering mutation questions, this is the format you'll use. You will remember this format exactly as it is, and you will just fill in the blanks. Remember it as I'm saying it to you now. Don't be stupid and not learn. Please don't do that for yourself, okay? Now, I'm just quickly going to carry on and say the following. Now, there's a change. The first thing you'll start saying is that when there's a mutation, firstly, there's a change in the nitrogenous base sequence of the DNA molecule from to thus resulting in a change in the codon from to okay? And as a result, different tRNA molecules carrying different amino acids are required to carry the code to the ribosomes. And provided that the amino acid changes from to or the sequence of the amino acid changes from to resulting in a formation of a different protein being formed. So it can be that the amino acid that is coded for is different. That would mean that the protein structure and function is thus then therefore different. But if the amino acid that we're coding for is the same, despite the change in the nitrogenous base sequence, we would still have the same protein. Okay? We would still have the same protein. But we'll get to that when we do more examples, boys and girls. I want us to quickly answer these questions. Let's start with question 2.1. Question 2.1.1 states the following. It says to us, we should actually identify the stage of protein synthesis shown here. They said protein synthesis at the ribosome. That would be, at the ribosome, that would be translation. 2.1.2, let's see. 2.1.2 says identify molecules X and Y. Molecule X and Y. That's translation, as we can see, because we said it's happening at the ribosome. Now, molecule Y. What would molecule Y be? I would say molecule Y is mRNA, and molecule X is tRNA. Why would I say that? I would say molecule X is tRNA because tRNA carries specific amino acids to the ribosome, not mRNA. You can see the amino acids are there, so therefore it's tRNA. Now, let's move on to question 2.1.3. It says state the term for the group of 3 nitrogenous bases indicated by V. Where's V? 3 nitrogenous bases indicated by V. And we see that V actually, we could see that V, let's see, it says the term for the group of 3 nitrogen indicated by V. V, we see that V is tRNA, right? And when V is tRNA, the 3 nitrogenous bases on tRNA would be called an anti-codon. Question 2.1.4. Now it says to us, give the nitrogenous base sequence, give the nitrogenous bases on the DNA strand that codes for UAU. So it says give the nitrogenous bases on the DNA strand, so the DNA strand, right, that codes for the bases UAU. So on molecule Y. So we see that molecule Y is mRNA. So molecule Y is mRNA, and we're looking to get to DNA. So thus, what would we do? They say that it's molecule Y, essentially. On molecule Y, the nitrogenous basis is UAU. So let's quickly write it down like this. Excuse me. I'm just going to write it off one beneath the other. UAU. So therefore, we're looking to go back to DNA. Now let's remember, what does DNA have? DNA has adenine, thymine, guanine, and cytosine. RNA has adenine, uracil, cytosine, and guanine. Fine. Now, what would adenine bond to when we go back? Adenine can't bond to another adenine. Thus, adenine would bond to a thymine. Uracil would not bond to uracil would not bond to thymine, but uracil would bond to adenine. So adenine and adenine. Now, does this make sense? We check ourselves. We're like, okay, when we were to follow the process and go forward, adenine would bond to uracil. Thymine would bond to adenine. Makes sense. Adenine would bond to uracil. So the answer, as we could see here, is ATA, essentially. The answer is ATA. 2.1.5. 2.1.5, it says to us, use the table below to identify molecule W. Where's amino acid W? So amino acid W is here, and it's UAU. So on the mRNA. So in the mRNA, it's UAU. And this table says it represents an amino acids on tRNA. So we don't have the tRNA sequence. So we just need to find it ourselves. So it's tRNA. And when moving forward, we see that, okay, uracil bonds to adenine. Adenine bonds to uracil. Uracil bonds to adenine. So now we're looking for AUA. Now what codes for you? What does AUA in tRNA code for? Let's see. Tyrosine. So that's our answer. So therefore it would be tyrosine. Tyrosine. That's your answer. And from there on, question 2.1.6 is something that is purely a memory question, which is 6 marks. So if you don't know this, or 5 marks, if you don't know this, buddy, you would be losing out. Learn the processes, learn the notes that I give you as a belief. Don't be stupid. Don't kill yourself. Okay? Don't shoot yourself in the foot. You're only disadvantaging yourself if you don't learn the notes that I give you, and you don't need another textbook. What you just need is to learn my notes. Okay? Now, question 2.1.6 says name. So when it says name, I want us to know that. Circle this, name. Key. So it means that's a mark for naming. And describe the process that occurs in the nucleus. What would occur in the nucleus? We said that transcription occurs in the nucleus and translation at the ribosome. So it's transcription. Name and describe the process that occurs in the nucleus to produce molecule Y. Where's molecule Y? produce molecule Y. What is produced? Molecule Y is what? As we said, molecule Y is mRNA. So how do we produce mRNA? So firstly, the process is called transcription. You get your compulsory mark. So you'd say firstly, DNA double helix unwinds, and then DNA double helix unwinds, and then what happens is that the weak hydrogen bonds between the DNA nitrogenous bases break, and when they break, essentially when they break, then the two strands unzip. One strand is used as a template, to form mRNA from free floating RNA nucleotides in the nucleoplasm. And then from there on, what would then happen is that mRNA is complementary to DNA. mRNA is complementary to DNA and obtains the coded message for protein synthesis, exactly as I've written it when we were doing